Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing Delhi University PhD entrance examination question paper for the subject genetics conducted in the year 2021. So coming to the first question. Shown are results of in vitro translation experiments using mRNA of a secreted protein with free ribosomes, that is lane 2, this one. That is around 50 kDa, and mRNA plus endoplasmic reticulum plus ribosomes followed by addition of Triton X100 at the indicated times after translation initiation, lane 3 to 7, that is from here to here. Then mRNA plus free ribosomes followed by addition of endoplasmic reticulum or salt washed endoplasmic reticulum 15 minutes after translation initiation, 8 and 9. These are the lanes. As control, secreted proteins from this specific mRNA is loaded in lane 1. This is the control, that is secreted protein. Answer the following question based on this data. Which statement best describes the protein product encoded by the mRNA? So, mRNA plus free ribosomes, they give a band at 50 kDa. But when Triton X100, that is over here, that is a detergent, that Triton X100 is a detergent, was added to endoplasmic reticulum plus ribosomes, there was no change in the 50 kDa band. That is from 3 to 7, there was no change in the 50 kDa band. And the final secreted protein, it had a band of 40 kDa. That means mRNA plus free ribosome produce protein, and endoplasmic reticulum with ribosomes does not play any role in the synthesis of protein. That is why treatment with Triton X100 to endoplasmic reticulum plus ribosomes, you will get a band of 50 kDa. Now mRNA plus free ribosomes followed by addition of endoplasmic reticulum, you get a band corresponding to the final secreted protein, that is 45 kDa. This shows that the protein is synthesized in the cytosol and it moves to the endoplasmic reticulum, where the amino terminal leader sequence of the polypeptide chain is cleaved by the endoplasmic reticulum protease. Now Triton X100 treated endoplasmic reticulum could not process the 50 kDa protein to 45 kDa, and so the 50 kDa band was obtained for endoplasmic reticulum plus ribosomes plus Triton X100. That is over here, because after Triton X100 treatment, it is a detergent and it will rupture the endoplasmic reticulum membrane proteins. Now mRNA plus free ribosomes plus salt washed endoplasmic reticulum, that is over here, also could not process the 50 kDa peptide, so the band appeared at 50 kDa in lane 9. Now which statement best describes the protein product encoded by the mRNA? So the mRNA encoded for a protein which is 50 kDa in size requires no processing within the endoplasmic reticulum. It shows that it requires processing within the endoplasmic reticulum. I have already given the explanation. Then the mRNA encodes for a protein which is 45 kDa in size in vivo. Not only in vivo, in vitro also it produces a 45 kDa protein. That is as per lane 8. Then mRNA encodes for a precursor protein which is translated in the cytosol and matures within the endoplasmic reticulum prior to secretion. That is correct. Option 3 is correct. Then next one, the mRNA encodes for a precursor protein which is translated on the endoplasmic reticulum bound ribosomes. That is not correct. It is taking place in the cytosol. So the correct answer is option 3. The next one, the translation of an mRNA encoding a secretory protein using a cell-free translation system containing microsomes, that is endoplasmic reticulum, lacking signal recognition particles, is initiated. Shortly afterwards, SRP molecules in the presence of triton, TX100 is triton X100, I told you it is a detergent, are added followed by further incubation. Which of the following outcome is the most likely? So the protein will begin synthesis on the ribosomes bound to endoplasmic reticulum. In normal case, that is in vivo condition, normally as the polypeptide emerges from the ribosome, signal sequences are recognized and bound by the signal recognition particle. But in this experiment, there is no SRP molecules to attach to the signal sequence. Shortly afterwards, SRP molecules are added in the presence of triton X100, a detergent. Now SRP molecules bind to the signal sequences on the polypeptide chain. Now here also I am going back to the normal condition. So in normal case, SRP binds the ribosome as well as signal sequence, inhibiting further translation and targeting this complex to the rough endoplasmic reticulum by binding to SRP receptor on the endoplasmic reticulum membrane. The polypeptide chain is then inserted into the membrane channel and the translation resumes. That is what is happening in normal condition. Now in the experiment, SRP molecules are added in the presence of triton X100. Now the microsome proteins, that is SRP receptor, will get separated from the microsome membrane due to the addition of triton X100. The SRP plus ribosome plus inhibited protein will not be able to bind to the SRP receptor and further synthesis of protein will not take place. So you will get a protein which is prematurely terminated. Now what will be the outcome? That is, protein synthesis will begin but will be terminated prematurely, leading to shorter products. That is correct. In all the others, it is the protein will be fully synthesized, which is not correct. Now coming to the next question. In a study of histidine biosynthesis in yeast, 6 new mutant haploids requiring supplemented histidine, that is histidine 1 to 6, this lane, in the medium for viability were isolated. The mutant haploids were fused in pairwise combination to form diploids. Okay. These are the mutant haploids. These are crossed in pairwise combination to form the diploids, whose requirement for histidine was tested. After crossing, they checked for whether the diploid requires histidine for its growth or not. The result of the test are shown below, where plus indicates diploid combination yielding histidine prototrophs. So prototrophs are organisms which grow even in minimal media. They are like wild types, not as auxotrophs. Auxotrophs will not grow on minimal media. So here I have made a what I can say for this. Okay, maybe a table like not a table actually, but a combination. I have just made it just for you to understand it very fast. Okay, so I have noted down which all require histidine. So in histidine 1, they require histidine because it is minus over here. That is, it doesn't grow in medium which does not contain histidine. So 1, then 1, 3 and 1, 6, they require histidine. Now here 2, 2, 2, 4 and 2, 5, it requires histidine. Then 3, 1, 3, 3, 3, 6 require histidine. Similarly for 4, 5 and 6, they all require histidine for their growth. Now those which do not require histidine, that is 1, 2. I have forgotten to put the 1 over here. Okay. It's 1, 2. This row is 1, 2. So 2 requires, then 4 does not require, and 5 also does not require histidine for their growth. Now if you take the case of this one, 2, 1, 2, 3 and 2, 6, it does not require histidine for their growth. Similarly 3, 2, 3, 5, 3, 4 does not require histidine for their growth. Similarly, in the next case also 136, that also does not require histidine for their growth. Then 2, 4 and 5 also does not require histidine for their growth. Now, now we see that when 1 is combined with 2 or 4 or 5, so the deficiency of lacking histidine 1 is overridden. Okay, that is in this case. But when 1 is crossed to 2, 4 and 5, the deficiency of lacking of histidine 1 is overridden. That is 2, 4 and 5 will help histidine 1 to grow in the medium. So even if histidine 1 is not added to the medium, the cells will grow if 2, 4, 5 genes are present. Similarly for 3 and 6, that is histidine 3 and histidine 6, 2, 4, 5 will be enough. Then when 2 is combined with, that is here, when 2 is combined with 1, 3 or 6, 1, 3 or 6, the deficiency of lacking histidine 2 is overridden. So even if this histidine 2 is not added to the medium, the cell will grow if 1, 3 and 6 genes are present. Similarly in the case of 4 and 5. So only 2 genes are represented among the 6 mutants. That is 2, 4, 5, 1 gene and 1, 3, 6. So answer here is option 2. 2, 4, 5 one set and 1, 3, 6 another set. So only 2 genes are represented. Then next one. You have 5 yeast strains each having distinct temperature sensitive allele of YFG1 and named TS1 to TS5 for impaired growth at 42 degrees Celsius. In the laboratory, you identify a suppressor to TS1 named SUP1 which restores growth at 42 degrees Celsius. Using pairwise crossing, you combine TS2 to TS5 with SUP1. It turns out that SUP1 when combined with TS2 to TS5 does not suppress impaired growth at 42 degrees Celsius. Which statement below best describes SUP1 function with respect to TS1? So you have 5 yeast strains and each having different temperature sensitive allele, that is YFG1 named alleles of YFG1 named TS1 to TS5 for impaired growth at 42 degrees Celsius. Now in the laboratory while you were doing some research, you identify a suppressor of TS1 named SUP1 which restores. Now TS1 it impairs growth at 42 degrees Celsius. So the suppressor, that is SUP1, it restores growth at 42 degrees Celsius. Now using pairwise crossing, you combine T2 to T5 with suppressor 1. So T2 with suppressor 1, T3 with suppressor 1, T4 with suppressor 1, T5 with suppressor 1. It turned out that suppressor 1 when combined with T2 to T5 does not suppress impaired growth at 42 degrees Celsius. So suppressor 1, it does not suppress the impaired growth at 42 degrees Celsius just like it had done to TS1. So to T2 to TS5 it is not a suppressor for impaired growth at 42 degrees Celsius. Now which statement below best describes suppressor 1 function with respect to TS1? So suppressor 1 is a dosage suppressor of TS1. So what is dosage suppression? It is a genetic interaction in which overproduction of one gene rescues a mutant phenotype of another gene. That is called as dosage suppression. Overproduction of one gene. Overproduction of suppressor 1 gene. It rescues mutant phenotype of another gene, that is TS1. Here suppressor gene is not overproduced. So it is not dosage suppression. Then interaction suppressors are compensating mutations in physically interacting components. A mutation in one protein disrupts the interaction, but a mutation in the second protein restores the interaction. Yes, that is correct. So suppressor 1 is an interaction suppressor because a mutation in one protein disrupts the interaction, but a mutation in the second protein, it restores the interaction. So if TS1 has impaired growth at 42 degrees Celsius, but when suppressor 1 is interacting with it, it restores growth at 42 degrees Celsius. So it is an interaction suppressor. Then bypass suppressor, it suppresses all mutant alleles of a gene including null alleles and hence bypass the requirement for a gene. That is called as bypass suppressor. And suppressor 1 is either a dosage or a bypass suppressor of TS1. So that is also not correct. So the answer is suppressor 1 is an interaction suppressor. So coming to the next question. Shown below are results of protease digestion reaction of sealed membrane vesicles derived from cells expressing membrane-bound protein MTG2P tagged with HA at the N terminus and with MYC at the C terminus. So which of the following statements best describe the localization of MTG2P? So they have asked how is MTG2P localized based on this gel data. How is the MTG2P, the membrane bound protein, localized? So they have tagged the N terminus of the protein with HA and C terminus with MYC. So the options are N terminus faces the cytosol and C terminus faces the lumen of the membrane vesicle. C terminus faces the cytosol and N terminus faces the lumen of the membrane vesicle. N terminus and C terminus face the lumen of the membrane vesicle. N terminus and C terminus both face the cytosol. Now the answer here is option 3, that is N terminus and C terminus both face the lumen of the membrane vesicle. Now we will see how. Now if suppose the N terminus and C terminus both are facing the cytosolic site, what will happen? Then the protein would have been denatured by proteinase K at any one of the concentration or at all the concentrations of proteinase K. Now this is This is the lane where proteinase K and Triton X100 are absent. So HA is showing a band like this and MYC is showing a band like this. That is the normal, that is the control conditions where you don't add both of these. But when you add at the proteinase K at 50 kDa without the addition of Triton X100, you get band for HA and MYC, similarly up till 200. But at 200 concentration, in the presence of Triton X100, you don't get a band for HA and MYC. Now we will see. When Triton X100 was added to the membrane, protein got isolated and this made the protein to be acted upon by proteinase K and it got denatured. So no band was obtained on the lane where both Triton X100 and proteinase K were treated, showing that both N and C terminus, that is N terminus is tagged with HA and C terminus with MYC, showing that both N terminus and C terminus, they are facing the lumen of the membrane vesicle. So the answer is N terminus and C terminus both face the lumen of the membrane vesicle. Now if they were facing the cytosol, then they would have got denatured by this time at different concentration of proteinase K itself, but it is not so. Only upon treatment with Triton X100, the membrane is broken and the protein N terminal and C terminal regions are exposed. So you don't get a band over here as proteinase K has already acted upon them. Then next one. You have a mixture of 3 proteins having molecular weights 40 kDa, 150 kDa and 250 kDa respectively. You separate them on size exclusion column packed in such a manner that proteins greater than 300 kDa elute in the void volume. What below best describes the elution order of the three proteins? Now we know that smaller molecules have more pores of the column material that is accessible to them. That is, they spend more time inside the pore relative to the larger sized molecule. Therefore, smaller molecules they will elute last and larger molecules they will elute first in the case of size exclusion chromatography. But here the condition is different because the size exclusion column is packed in such a manner that proteins greater than 300 kDa elute in the void volume, whereas the others they enter into the pores of the column material. So molecules that enter the pores will have an average residence time in the particle average residence time in the gel particle that depends on the molecule's size and shape. So now size of the pore of the column will be less than or equal to 300 kDa. Okay. So 250 kDa molecule will occupy the maximum space in the pore when compared to that of 40 kDa and 50 kDa. Since it has occupied maximum space in the pore, its movement through the pore will be slow, so it will elute last. Whereas the 40 kDa protein is very small in size compared to the size of the pore, so it will elute first. 150 kDa is between the size of 40 and 250 and will elute the second. So the answer here is 40 kDa followed by 150 kDa followed by 250 kDa. So the answer is option 1. Then next one. Mitochondrial membrane fractions were treated with either a tris HCl buffer pH 8 or a tris Cl buffer pH 8 containing 1 molar NaCl. Soluble and pellet fractions were separated by centrifugation and probed for the presence of MTG2P. Shown below are the results. Now, tris HCl with an effective pH range of 7 to 9 is capable of extracting soluble cytoplasmic proteins. Now, peripheral proteins. Peripheral proteins, they are associated with the membrane by weak electrostatic bonds, and these are usually solubilized by extraction with high concentration of salt solution that weaken the electrostatic bond holding the peripheral proteins to the membrane. Now, MTG2P, it is a peripheral membrane protein, and a treatment with the tris buffer alone is not sufficient. So that is why it did not appear in the soluble fraction and appeared in the pellet. That is over here. This is the first. It did not appear in the soluble fraction, but it appeared in the pellet. But on addition of NaCl, here in this case, but on addition of NaCl, the peripheral membrane protein got separated and entered the soluble fraction. That is why you got a band over here. So the answer here is MTG2P is a peripheral membrane protein. Peripheral membrane proteins, they are usually isolated using high concentration of salt solution that weaken the electrostatic interaction holding the peripheral proteins to the membrane. Then next one. The following experiment was carried out to identify the different regulatory elements present in a promoter. 3 different mutated promoter X, Y and Z were developed where different regions were mutated as indicated by filled regions in the figure below. The promoter was cloned upstream to a reporter gene and the activity of the mutated promoter studied in cell line. The wild type promoter was used as control. The activity of the wild type and the mutant promoters are mentioned in the figure below. So, the answer is 2, that is in promoter X, the binding site for RNA polymerase is mutated. In promoter Y, binding site for a negative regulator is mutated, and in promoter Z, binding site for a positive regulator is mutated. Now we will see how. So in X, that is over here, in X, normal polymerase binding site is mutated, okay. Is mutated. So transcription translation of reporter gene doesn't happen, so it shows 0 activity. Wild type is having 80% activity. Whereas this one where the promoter itself is mutated, you have 0% activity. Then Y is the binding site for the negative regulator, that is over here, negative regulator or the repressor. So when that region is mutated, the reporter gene showed 100% activity. That is how you got this particular answer. Then Z is the binding site, that is this site is the binding site for a positive regulator or the activator. That is why when it got mutated, you got only 60% activity, less than 80% in wild type. So that is the reason for answer 2. So coming to the next question. Mendel in his experiment observed that round seed phenotype was dominant to wrinkled seed. Okay. In order to distinguish between homozygous and heterozygous round-seeded plants in the F2 progeny, he had selfed each plant and analyzed their progeny. So he has done breeding experiments. Now fast forward to 1990. Scientists had identified that insertion of a transposable element leads to wrinkled phenotype. So scientists who were working in the 1990s, they identified that transposable element was responsible for wrinkled phenotype. When the genes responsible for round and wrinkled seed shape is isolated and run on a gel, the pattern observed is as shown in figure A. So this is the gene that represents, this band represents smaller and this band represents capital R. Then in 2016, again fast forward, first it was in 18s, then it was in 19s, and now it is in 20s. So in 2016, a geneticist crosses two pea plants and screens the progeny for gene responsible for round and wrinkled seed shape. The profile of DNA bands obtained are shown in figure B. The number of progeny with each profile are also indicated on the top. So after crossing, you get a band that is corresponding to smaller as well as capital R in the same lane. Then you have got a band corresponding to capital R in the next lane. Now they have given so many crosses and dominance and codominance here in the options. So if it was a dominant marker, now suppose if it was a dominant marker, then you would have obtained only a single band as shown here. That is this picture which I have drawn. So in this picture you would have got only a single band which corresponds to capital B. And that band would have corresponded with the band in lane 3 of this particular picture, that is for heterozygous condition if it was a dominant marker. Now if it was a codominant marker, then you would have got a band for in the F1 you would have got a band for capital A corresponding in this picture which I have drawn, corresponding to capital A as well as small a for this heterozygous condition that shows its codominant nature. So we will rule out the dominant options and we will take only the codominant ones. Now in the cross between capital R into smaller, you will get F1 as capital R small r. You cross it, you get F2, and F2 progeny will be capital R, capital R, capital R small r, capital R small r, and small r small r. But here you are getting only two types of progeny, that is capital R small r and capital R capital R. So it should be this, that is fourth option, capital R small r into capital R capital R. You will get progeny, that is capital R and capital R small r. So the answer here is option 4, that is R capital R small r into small capital R and codominant. Next one, mammalian cell cultures were treated with chloramphenicol for 30 minutes prior to addition of S35 methionine. What molecular entity will not be labeled? So chloramphenicol and tetracycline, they can inhibit mammalian mitochondrial protein synthesis. So proteins in mitochondria will not be labeled. Answer is option 4. Then a cell lysate was solubilized in presence of non-ionic detergent and treated with EndoH, which cleaves N-linked glycosyl moieties. Shown below are Western blots with antibodies to integral membrane protein IMP1 containing multiple N-linked glycosyl moieties in the absence or presence of EndoH. Which statement below does not accurately describe the cellular localization of IMP1? So EndoH, without EndoH treatment you get a band, with EndoH treatment also you get a band for IMP1. Now when linked glycosylation, it occurs co-translationally and is initiated in the endoplasmic reticulum. Now if it was an endoplasmic reticulum protein as in option 1, in the presence of EndoH you would not have got a band corresponding to the one in which EndoH was not added. So the answer here is below does not accurately describe the cellular localization of IMP1. It is IMP1 is an endoplasmic resident protein. The other option, that is IMP1 is a Golgi resident protein. Yes, it can be. IMP1 traffics from endoplasmic reticulum to Golgi and back. Yes. IMP1 is plasma membrane protein. That is also correct. But option 1, it is not correct. And that is your answer. Then next one, on discovery of a virus with the circular double-stranded DNA of approximately 10,000 base pairs, its map was constructed by digesting the DNA with various restriction endonucleases. The following results were obtained. So ECO-R1 gave 2 bands, HIND3 gave 3, BAM-H1 gave 1, ECO-R1 HIND3 gave 1, 2, 3, 4, 5. ECO-R1 BAM-H1 gave 3, HIND3 BAM-H1 gave 4, and ECO-R1 HIND3 BAM-H1 gave 5. So we will see for the triple digest first. ECO-R1 HIND3 BAM-H1, this one. So it is 3.3, 1.8, 1.5, 1.1 and 0.5. So if you see option 3, that is this one, you see that ECO-R1 HIND3 and BAM-H1. If you digest with ECO-R1, you will cut over here. You can get 1.1, 0.5 and 1.5. These bands appear over here. But in between HIND3 is there, so they will also digest between the ECO-R1 digested fragments, giving rise to 1.1, 0.5 and 1.5. That is over here, 1.5, 1.1 and 0.5 band. Then comes HIND3 again, that is over here. It will digest and you will get a 3.3 fragment. So that is also been there. Then BAM-H1, it will digest between HIND3 and ECO-R1 fragment and you will get a 1.8 fragment. That is also here. So the answer here is option 3. You can even cross check with the double digest and single digest also. It will be corresponding to 3. Answer is option 3. Then next one. You wish to amplify the region of caps below using PCR. Select the correct set of 8 base pair primer pair. Okay. So this is the sequence. This is the sequence. I have rewritten it over here in 5 prime 3 prime direction. This is the complementary sequence to that. This will be your forward primer, this one. So you start from here, A, G, G, A, C, T, T, A. That is option 1, that is A, G, G, A, C, T, T, A. And this complementary to the second strand will be your forward primer in the 5 prime 3 prime direction, and it will be similar in sequence to the first strand, that is this one, in the 5 prime 3 prime direction, that is A, G, G, A, C, T, T, A. Now see for the reverse primer. Reverse primer will be the complement of the first strand, but in this direction, that is 5 prime to 3 prime direction, in this way. So it will be G, 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 C, C, A, G, T. That is your reverse primer. So it is as per given in option 1. So the answer here is option 1. Then next one, you want to express recombinant human insulin protein in E. coli. You are given a clone containing the pig insulin gene and human pancreatic tissue. Lists below are the steps that are required. So choose the correct answer from the options below. So isolate mRNA from human pancreas. That would be the first thing that you have to do. Then using reverse transcriptase make cDNA, that is D. Then comes clone the cDNAs into expression vector to make library, that is F. And then probe cDNA library with cloned pig insulin, that is B. Then select positive clones that hybridize to the cloned pig insulin gene, that is C. And express recombinant human insulin in E. coli, that is E. So the answer is option 4. A, D, F, B, C, E. The next one. A PCR reaction that continues for 30 cycles will produce approximately how many PCR products from a single template DNA molecule? So after 30 cycles, as many as a billion copies of the target sequence are produced from a single starting molecule. So approximately 1 billion. Answer is option 4. Then next one, for 96 MCI radioactive sample with half-life of 2.7 days, how many days later will be 6 of the sample be left? So here you have 0 days, that is 96 MCI of the radioactive sample is present. After 2.7 days it get reduced to half, that is the half-life of that particular sample, and it is 32. Next 2.7 days it will become 16. Next 2.7 days it becomes 8, and next 2.7 days it becomes 4. So 4 is near to 6. So that will be after add all the 3. 1, 2, 3, 4. Or add all the 4 and you get 10.8 days. So the answer here is option 4, 10.8 days. Then next one. A plasmid is digested with EcoR1, GAA-TTC to linearize it and at a cloning site, and a DNA fragment is digested with KPN1. For cloning, both the vector and the DNA fragment is treated with clinoenzyme and 4-DNTP substrate to make them blunt ends, following which they are ligated. Which of the following represent the sequence of ligated junction at the point of ligation between the blunt-ended EcoR1 and KPN1? So, you have digested the plasmid with EcoR1 and the gene of interest with KPN1, and then you have treated both of them with clinoenzyme and DNTPs, and you have made blunt ends, and which following which they are ligated. Then which of the following represents the sequence at the ligated junction? That is the question. So this is the plasmid. It is having this E carbon site. This is all just for understanding. I have written it like this. Okay. This is the plasmid with the GAATTC. So that is the 5' 3' direction, and this one in 5' 3' direction will be like this. It will make cuts over here, that is at G, and you will get something like this with 5' overhangs G. Okay, you will get something like this with 5' overhangs. Then you have got the your DNA of interest, which is flanked on both the sides with KPN1 site. So this is the KPN1 site, and here also KPN1. Then you digest with KPN1, you get 3' overhangs, that is over here, and here get 3' overhangs. Then you treat both of them with Clino. So this plasmid, upon treatment with Clino, it will show 5' 3' polymerase activity that fills in 5' 3' direction. So this region will be filled, and you will get GAATT, and here you will get AATT like this, and you get a blunt ended plasmid. Now similarly, if you treat with Clino for the DNA of interest, 3' 5' exonuclease activity of Clino, it will cleave the 3' overhangs. So this region will be cleaved, this region also will be cleaved, and this region also will be cleaved, and you will get something like this. And with this gene of interest of yours, then you ligate both of them, that is like this. So you will get GAATT and this C, and G from here, and AATTC from here, and CTTAG from here, and CTTAAG from here. C first coming from this one. And this is your ligation product. And what they have asked, which of the following represent the sequence at ligated junction? So at ligated junction, the sequence is GAATTC in the 5' 3' direction. Here also GAATTC. So the answer is option 2. I hope you all understood this. This is only for your information. I have made this picture for you to understand easily. GAATTC. Then next one. Among the statements below, which least describe the nature of mutagenic mechanism by nitrous acid and 5-bromouracil? So nitrous acid and 5-bromouracil act only on replicating DNA. Answer is option 3. Then next one. Inactivation of mutation of which gene within E. coli lactose operon would lead to its constitutive expression? So if operator is constitutive mutant in case of lac mutants of E. coli. So the answer here is operator. Answer is option 3. Then next one. You want to make 100 ml solution of 0.1 normal MgCl2. You are provided with 1 normal stock solution of MgCl2. The volume of water and the stock MgCl2 that will be needed to be mixed is M1V1 is equal to M2V2. So 1 into V1 is equal to 0.1 into 100. So V1 will be 10 ml. So 10 ml MgCl2 and 90 ml water to make 100 ml solution. So the answer here is option 1. Then next one. Given that husband and wife are both heterozygous for a recessive allele for albinism, if they have dizygotic twins, dizygotic means fraternal twins, not identical, the probability that both the twins will have the same phenotype for pigmentation will be. So this pigmentation, it is an autosomal character. So capital A small a and capital A small a, as both husband and wife are heterozygous for a recessive allele for albinism, and these are the offsprings that they will obtain Then if they have dizygotic twins, the probability that both the twins will have the same phenotype for pigmentation will be. So probability that the first twin will be pigmented is 1 out of 4. This one, 1 by 4. Because it is a recessive allele, this one small a is considered. So 1 out of 4. Probability that second twin will be pigmented is also 1 out of 4. Probability that both of them will have the same phenotype, that is both of them will be pigmented, is 1 out of 4 into 1 by 4. So 0.0625 is the answer. Answer is option 4. The next one. If the recombination frequency between XY locus is 12, XZ locus is 4, YZ loci is 8, then the order of the loci of the chromosome is. So they have given XY is 12. So if you mark X and Y at the ends of this particular line, that whole thing will be 12. And 4 plus 8 also will be 12. So YZ loci is, that is over here, this is 8, and XZ loci is 4. So the order will be X, Z and Y. Option 3. The next one. The genotypes of husband and wife are IA, IB, IAI. Among the blood types of their children, how many different genotypes and phenotypes are possible? So for convenience, I haven't written I capital A, I capital B, etc. I have just written AB and AO. Now these are the gametes produced by AB and this is by AO. Now these are the offsprings that they will be obtaining. Now how many different genotypes will be there? 1, 2, 3 and 4. 4 genotypes. And how many phenotypes? This is A blood group. This is also A blood group. This is AB and B. So 1, 2 and 3 phenotypes. So the answer is 4 genotypes and 3 phenotypes. Answer is option 4. So coming to the next question. For a given genotype capital A, a diploid cell at metaphase will have. So it will have 2 copies of the alleles on 2 chromosomes. Sorry, 4 copies of the alleles on 2 chromosomes. Answer 2 is the answer. Then next one. You transcribe mRNA from DNA sample and purify it. You then separate the 2 strands of the DNA and analyze the base composition of each strand and that of the mRNA. You obtain the data shown in the table below. Which strand of the DNA is serving as template for mRNA synthesis? So this is DNA strand 1. So this is the composition of ATGC. Sorry, in DNA strand 1, and this composition of ATGC is similar to, or I can say it's almost same as that of the mRNA strand. So that is the sense strand, because sense strand and mRNA are the same except that T is replaced by U in mRNA. So that is then sense strand or coding strand or non-template strand. Now the template strand will be DNA strand 2. So they have asked for the template strand, so that is strand 2. Answer is option 2. The next one. A mutant of E. coli with heat sensitive DNA ligase 22 degrees Celsius permissive, 37 degree restrictive have been used to show that the DNA synthesis is discontinuous. Examination of DNA replication in the presence of H3 thymidine in the mutant would demonstrate the accumulation of short fragments of. So as per the question, they have asked for accumulation of short fragments, and this can happen only at restrictive temperature, that is 37 degrees Celsius, and not at permissive temperature, that is 22 degrees Celsius. Why? Because at permissive temperature, DNA ligase would have joined the fragments. So answer is option 3, that is radioactive DNA at 37 degrees Celsius but not at 22 degrees Celsius. Then next one. Which one of the following histones is required to achieve the solenoid structure of chromatin? So it is histone H1. Option 1 is correct. Histone H1, it plays a role in stabilizing the 330 nanometer solenoid structure. The long N-terminal and C-terminal ends of the H1 protein attach to adjacent nucleosome core particles, pulling them into an orderly solenoid array. Then next one. Which of the following proteins is required for establishment of monopolar attachment of sister kinetochores during meiosis? So during meiosis, sister chromatids produced by chromosome duplication are held together by REC8, a meiosis-specific cohesin protein. And REC8-like protein are required for mono-orientation of sister chromatids during meiosis 1 in plants and in fish and yeast. So the answer here is option 1. Then which of the following parts of the brain function as the primary respiratory control center? It is medulla oblongata. Answer is option 2. Then which one of the following disorder manifests due to defects in nucleotide excision repair? It is xeroderma pigmentosum. Answer is option 2. Then which of the following is a bypass repair system? It is SOS repair system, that is bypass. Answer is option 3. Then the spatial distribution of mRNAs in a cell can be detected by. It can be detected by fluorescence in situ hybridization. 4. Then matrix assisted laser desorption ionization time of light multi-top spectrometry is most useful for predicting which of the following? It is used for predicting molecular mass. Answer is option 3. Then which of the following techniques cannot be utilized to demonstrate protein-protein interaction? So it is yeast 3 hybrid assay. It is for protein RNA interaction. So answer is option 3. The next one. Cyclins facilitate progression of cell cycle by. So activating the protein kinases which are critical regulators of cell division. Answer is option 1. Then translation repression does not include. So it does not include decapping enzymes. Answer is option 1. Then a patient has an abnormal karyotype exhibiting 3 copies of chromosome 21. This chromosomal anomaly most likely arose from an error during the following stages of cell cycle. So 3 copies of chromosomes 21 is Down syndrome. It is trisomy of chromosome 21. And Down syndrome is caused by the presence of an extra chromosome number 21. Both the chromosomes and of the pair 21 pass into a single egg due to non-disjunction during oogenesis in the mother's ovary. Thus, the egg has 24 chromosomes instead of 23 and the offspring has 47 chromosomes instead of 46. So answer is option 1, that is meiosis 1. Non-disjunction during meiosis 1. The next one. The development of the anterior-posterior axis of Drosophila is initiated when? So, the mother contributes and packages bicoid and nanos mRNA into developing oocyte. That is correct. The anterior-posterior axis of Drosophila is initiated then. Answer is 1. Then next one. Plasmid vectors used in cloning often contain a gene for N-terminal 146 amino acids of the enzyme beta-galactosidase. What is the purpose of including this gene in the vector? So this is a screen for recombinant vectors with inserts. XGAL IPTG medium, similar to that. Contain a gene for N-terminal 146 amino acid of the enzyme beta-galactosidase. What is the purpose of including this gene in the vector? It is used to screen recombinants with inserts. Then, the unusual property of Taq polymerase that is critical to the PCR is its. So is it thermostability? The Taq polymerase is highly thermostable. Answer is 2. Then which of the following amino acids are incorrectly grouped based on their side chain polarity properties? So the answer here is phenylalanine, tyrosine and tryptophan. So phenylalanine and tryptophan, they are hydrophobic and tyrosine is polar. So answer here is option 3. Now this is a mistake, I think. Don't see this one. It is been repeated from here. Okay. Which of one of the following listed processes below starts from 3' to 5' direction? So it is mRNA editing. Answer is option 4. I have discussed on mRNA editing in I think set exam papers, either Maharashtra or Karnataka or West Bengal. These three I have found mRNA editing questions being asked. You can refer to them also. Then next one. Access of transcription factors to DNA is usually influenced by. So it is by acetylation of histones in the euchromatin. So acetylation of histone, it alters the accessibility of chromatin and allows DNA binding proteins to interact with exposed sites to activate gene transcription and downstream cellular functions. And sumoylation, it is a post-translational modification. So answer is option 1. Next one. This amino acid is not yet found in proteins. So it is D-lysine that is not yet found. L-lysine is found in nature than D-lysine. So answer is option 3. Then the protective mechanism in a eukaryotic cell that destroys mRNA leading to its reduced expression via a double-stranded RNA with the same sequences. It is known as RNA interference. Answer is option 2. When a culture of bacteria is shifted to high temperatures, the heat shock response is triggered by. So immediately upon temperature upshift, the cellular level of sigma 32 responsible for transcription of heat shock protein increases rapidly and transiently. So the answer is specific sigma factors. Answer is option 2. The next one. RISC mediates all of the following aspects of RNA interference except. Now RNA interference, it is a post-translational gene silencing method, and the mechanism by which RNAi works is initiated by progressive cleavage of the double-stranded RNA into 21 to 23 nucleotide siRNAs. These siRNAs are then incorporated into protein complex called RNA-induced silencing complex or RISC, and ATP-dependent unwinding of the siRNA duplex generates an active RISC complex, active RISC complex. Guided by the antisense strand of siRNA, the active RISC complex recognizes and cleaves the corresponding mRNA. So RISC mediates all of the following except binding double-stranded RNA, yes. Chopping up double-stranded RNA, no. Degrading mRNA, yes. Unwinding double-stranded RNA, yes. So answer is option 2. Then next one. An alpha helical conformation of a globular protein in solution is best determined by which of the following? It is circular dichorism. Answer is option 2. Then which is the common terminology used for enzyme which activate or inactivate other proteins by phosphorylating them? So it is kinase activity, protein kinase. Answer is option 2. Then you are testing a new plant product A as potential new therapeutics in cultured human cells to arrest the cell division at this phase of the cell cycle. Which of the following is the most likely a target of molecule A? So it will be topoisomerase. Because topoisomerases, they are or I can say topoisomerase inhibitors can inhibit cell proliferation and induce cell cycle arrest by preventing DNA replication. So topoisomerase poisoning or inhibition leads to replication fork arrest and double-stranded break formation and inducing cell cycle arrest. So the answer is topoisomerase. So that's it. Thank you for watching my channel. Please do like, share and subscribe. Thank you.